Hello, welcome to my YouTube channel, The World of Science. Today we will discuss an important topic of microbiology, which is microbial identification. Uh, Today we discuss traditional method, and in another next video we will discuss modern method. In traditional method, we have culturing, staining, biochemical test, and substrate utilization test. We will discuss these techniques in detail. Before we discuss their types, we must know where it is used to. So, main use for accurate microbial identification are in healthcare, epidemiology, and pharmaceutical industry, and also in number of departments. In healthcare, accurate and fast identification of bacteria, fungi, and parasite for correct and timely disease diagnosis and appropriate treatment. In epidemiology, for the tracking and tracing of disease, spread and outbreak, as well as the identification of new isolates, for example, antibiotic-resistant isolates, as well as we can see that microbial identification is also used in pharmaceutical industry because microbes are a significant threat to sterility. Accurate identification of environmental microbe is often a good manufacturing practice, which we usually call GMP, which required which is a requirement within the pharmaceutical industry. Additional scenarios including criminal investigation, microbial forensic, and environmental studies. We also use microbial identification. So we discuss traditional vs modern method. Traditional methods for microbial identification rely only phenotypic identification using staining, culturing, and simple biochemical tests. As you know that phenotype is a set of observable characteristics of an individual, like physical size, shape, color, smell, etc. Nowadays, newer and more powerful molecular, immunological, and biochemical analytical methods support and sometimes replace the traditional method. In modern method, we usually use molecular, immunological, and biochemical methods. While in traditional method, we use simple biochemical tests, culturing, and staining, etc. So, so we discuss broadly in what is traditional method and what are the different types of traditional method for microbial identification. Before studying traditional method, I will like to mm, mention you microscopic feature of traditional method. Microscopic feature encompass the overall appearance of growth of an organism, usually colonial appearance, include its shape, size, color, and smell. By examining the gross microscopic feature on an agar culture, the type of microorganism can be usually determined. As you know that agar is a solid gel-like base used for most of the solid media. So here you can see the petri dish with agar in which usually bacteria, fungi, and other microorganisms usually grow. So we discuss one by one uh, in agar culture. So a general guide to examine agar culture. First we discuss fungi. Filamentous fungi are mold, usually appear as a hairy, irregular shaped colonies, and often produce visible spore that may look powdery or dusty. So fungal colonies can contain more than one color, usually with a darker color in the center and a lighter color radiating from the center. Filamentous fungi grow radially from the center of an agar, with the youngest on the outside and the older, darker material on the inside. This is the diagram, a picture of the agar culture of fungi. You can observe it. Filamentous fungi may also grow as a unique colored pyramid with no obvious sign of spore, as it is shown in the diagram. So after fungi, we'll discuss bacteria. We uh, we discussed that how bacteria look in agar culture, what is their color, odor, and smell, etc. So bacteria often form distinct colonies. These colonies are usually smaller than fungal colonies. Uh, in texture, they can vary from slimy to wet dry. They can be of any color, but colonies of any species will always have same color and appearance on a particular culture media. Like you can see here a number of uh, bacterial culture with different shape, size, color. So bacteria have a strong odor, with filamentous fungi can be odorless on earthy smelling. This is the one of the important point that can we use to differentiate bacteria and filamentous fungi, because they are in appearance they look very similar to each other. So if we smell their odor, then the bacteria have strong odor, while filamentous fungi can be odorless or earthy smelling. Yeast can be trickiest to identify based on microscopic feature, because colonies can often look similar to bacterial colonies depending on the species and type of agar used. Here is the diagram of bacterial colonies on the left side and the yeast colonies on the right side. So the second method is the staining. First we discuss what we discuss. First we discuss culture. The second one is staining. So staining most often researcher applies stain to microbial sample for easier visualization under a microscope. There are number of staining. Uh, Few of them we discuss here. Number one is gram staining. Gram staining is often the first test performed for bacterial identification. Why it called gram stain? This purple stain based on crystal violet dyes is named after the Danish bacteriologist Hans Christian Gram, who developed it in 1884. Usually gram stain we use to identify bacteria, whether it is positive gram positive bacteria or gram negative bacteria. So typically gram-positive bacteria include Bacillus, Staphylococcus, Streptococcus, and Clostridium species. And typically gram-negative bacteria include Escherichia, Helicobacter, and Salmonella species, etc. Certain bacteria are gram-variable, and they are therefore not suitable for identification of gram staining. Here is the microscopic appearance of a gram-positive bacteria and gram-negative bacteria. The left one is gram-positive and the right one is gram-negative bacteria, which usually is seen through microscope. After gram staining, another staining technique is one of the important staining technique is endospore staining. This technique involves applying a stain to a bacterial sample to check for the presence of a spore. Because not all bacteria produce spore, this information can be useful in identification. Several spore strains are available, and Malachite green is probably the most popular one, which we use in endospore staining for the identification of microbe, whether it's a endospore spore producing microbe or bacteria or not. Here is the diagram. So the third one is Zeal-Nielsen staining. It is one of the it is also one of the most important staining method techniques. This is an important tool for staining of Mycobacterium tuberculosis, which we usually call TB, which cannot be gram stained. The red stain carbon fustation is used and followed by a contrast stain such as methylene blue. M tuberculosis stain red while others stain blue. So through microscope we can see such like the diagram. The red stain is a Mycobacterium tuberculosis, while the blue stain are methylene blue. So the gram staining, Zeal-Nielsen staining, and interspore staining are usually we used for identification of bacteria. So for fungi and yeast, several strains exist, although these are generally non-specific in nature. They help visualize fungal element for identification rather than differentiate between fungal species. Example of fungal stain include lactophenol cotton below stain, which stain carbohydrate present in the fungal cell wall. So we easily visualize the fungal element, which is carbohydrate in their cell wall. Similarly, this is diagram. Uh, Similarly, periodic acid shift stain, which is also called base stain. This stain, this stain carbohydrate in other moieties and led to a magenta, means a purplish red color in living fungi only. This is used in infection disease. Usually, it is used for diagnosis of infection. So this is also common one in biochemical labs. After this, tryptophan blue, aniline blue, and chlorophyll white are other commonly used fungal stain in labs and used for the identification of fungi and yeast. As we discuss uh, spore agar culture, staining and staining method for identification of microbes. The third one is a catalyzed simple biochemical test, which include catalyzed test, oxidase test, etc. What is catalyzed test? If the unidentified bacterial species has catalyzed activity, bubble of oxygen appeared when hydrogen peroxide is added to a scraping of bacteria on a microscopic slide, like Staphylococcus, Mirococcus, E. coli, and Salmonella species are amongst the most routinely encountered catalase positive bacteria, while the Streptococcus and Enterococcus bacteria are catalase negative. So they produce bubble of oxygen when hydrogen peroxide is added. In this way, we determine whether it is positive or negative. Uh, Here is the diagram. The second one is oxidase test, which we usually use in simple biochemical test. The oxidase test identifies bacteria with cytochrome C oxidase activity because there are number of bacteria which have cytochrome C oxidase activity. So in this way, we can identify those bacteria which have cytochrome C oxidase activity. This, as we know that this enzyme form a part of bacterial electron transport chain. When present, ca- cytochrome C oxidase oxidize the reagent to a purple color product. In this way, we can identify that the purple color product determine, show that this is the bacteria which have cytochrome C oxidase activity. When the enzyme is not present, the reagent remain in the reduced state and is colorless. While oxidase positive bacteria are aerobic, they are not necessarily strict aerobes and may be capable of anaerobic respiration. Similarly, a false negative result is obtained if bacterial species possesses an oxidase incapable of first reacting the test reagent. So the fourth one is the substrate utilization test. A range of substrate utilization tests for microbial identification is available commercially. These tests, usually for bacterial species, contain a panel of substrate, example, different carbon and nitrogen source. Bacterial species use these substrate differentially. Fermentation tests using various sugar are important. During fermentation, acid is produced which is indicated by change in color of an indicator. Here you can see negative carbohydrate fermentation, which is red, and positive carbohydrate fermentation, which is yellow, and fermentation and gas production, which is positive, which also positive, which is somewhat light yellow color. A record of the color change in substrate after incubation with bacteria generate a key or pattern of substrate utilization. This key is compared to the substrate utilization pattern of a computerized list of bacterial species. These tests are probably more reliable in combination with kitless oxidase testing and microscopic examination. These tests overlap with the concept of selective growth media. A wide range of selective media exist for the isolation and identification of bacterial and fungal species. Here is McConaughey agar. You can see that left lactose fermenting pink colonies, while the right one is non-lactose fermenting. So these are the most of some of the most important traditional methods which usually used in different clinical laboratories. Uh, so thank you for watching, and must watch my next video, which is based on modern method of microbial identification. Thank you.